Okay, so today's video is going to focus on kind of the second half of photosynthesis. The last set we covered all the light reactions and the transfer of energy there, the formation of the ATP and the NADPH. So your last set of videos kind of covered this section here, okay, where the light energy is coming in and that's going through the electron transport chain, and it's helping form that proton gradient that will then make ATP and also as well as will make the NADPH that's used in the part we're going to talk about tonight, which is the Calvin cycle over here. And so the Calvin cycle is where that chemical energy, because remember the light energy here, this diagram here is energy, so the light energy here has been converted into chemical energy. Remember the energy is neither created nor destroyed, it's just changed. Okay? And so that chemical energy, okay, is going to produce organic compounds, which is also essentially another version of chemical energy. And so the Calvin cycle, remember it occurs in the stroma of the chloroplast. And remember the stroma is the liquid portion of the chloroplast, okay, surrounding all those thylakoid membranes, on the outside of the thylakoid membranes. And so that's where our focus is going to be tonight. So this is kind of a two-step process here, okay, in getting the carbons out, okay, and out of the Calvin cycle to be able to form the organic compounds. So we're going to go through carbon fixation as well as reduction, and then we'll complete the cycle. Okay, so our first step here in this in the Calvin cycle is going to be this carbon fixation. Okay, and so carbon fixation is when I'm going to take the carbon from a non-organic compound. An example would be CO2. Okay, so I'm going to take the carbon, or I'm going to, a lot of times you'll see, they'll use assimilate. Okay, so I'm going to assimilate the carbon from a non-organic compound. And I'm going to take that carbon and I'm going to incorporate it into an organic compound. Okay, so I'm going to assimilate the carbon from this non-organic compound into this organic compound, okay, which is going to be some kind of carbohydrate. A lot of times it is glucose, but it does not always have to be glucose. So our carbon fixation is going to happen when the molecules of CO2 from the atmosphere, okay, so that's these guys over here, and they're in red because these are the molecules that have come from the atmosphere. So these are the molecules that came from my inorganic compound. Okay, they came from the CO2 in the atmosphere. So I've got 3 CO2 molecules, okay, that are joined to 3 molecules of what's called RUBP, okay, ribulose bisphosphate. You don't have to know its name. Okay, you just need to know RUBP. So RUBP is a 5-carbon sugar, okay, and you can see, if you count your carbons here, okay, I've got 5 carbons in each of these molecules of RUBP. Okay, so it's a 5-carbon sugar, and it uses the enzyme Rubisco, okay, to join these together. Okay, so this is the carbon fixation process here. Okay, is I'm adding these carbons from my inorganic molecules to my RUBP. So this is going to end up as 6-carbon molecules because I'm going to add them together. And Rubisco is the enzyme that's going to do this. So like I said, I'm going to end up with 3 molecules now that are 6 carbons long. So I'm going to have a total of 18 carbons, where before this carbon fixation process started, I had 15 carbons because I had these three 5-carbon ribulose bisphosphate molecules. And now after carbon fixation, I'm going to end up with 18 carbon molecules. So my second step here in the Calvin cycle is going to be the reduction process. And you can see here, I've got my 3 6-carbon molecules that were done. These are the result of the fixation. Okay, you can see my red carbon that came from the CO2 attached on the ends here. Okay, so these were originally the RUBPs, okay, and my original RUBP molecules down here, okay, and then my 6 carbon here that came from the CO2. Now this molecule, okay, these molecules are going to go through the process of reduction. They're going to get split in half, forming 6 3-carbon molecules, which you can see over here. Okay, so I've got 6, 1, 2, 3, 4, 5, 6 3-carbon molecules, okay, over on the side there. And then they're also going to be reduced. Okay, remember when we reduce something, okay, I'm reducing, okay, the positive charge. And so I'm going to reduce these molecules. You can see my NADPH right here has gone back to being NADP+, okay, because that NADPH, remember it's an electron carrier? That electron got excited in the photosystem and it went down the electron transport chain, and NADPH was the final electron acceptor, and it was so that it could bring that electron over here to the Calvin cycle. And so now this NADPH is going to donate its electron. It's going to reduce these molecules. So these molecules are going to split, okay, into these 3-carbon molecules. Okay, they're going to be reduced by the NADPH, and so now I have 6 3-carbon molecules that are called G3P or pGal. Okay, that's this right here. So that's the name for all these guys up here. So they're called G3P or pGal. Okay, so let's look at what I have now. I've gone through fixation and I've gone through reduction. So remember these molecules over here, okay, these are my pGal or my G3P. Remember, they're the same thing. I've got 6 of those molecules. So let's talk about the amount of carbons we had. So now I've got 18 carbons. And remember, when I started this whole Calvin cycle, I only had 15 carbons. Okay? Well, we need to think about the fact that this is a Calvin cycle. So if I'm going to go through this cycle, I've got to get back to the beginning. And at the beginning, I had 15 carbons. And now I'm over here in the cycle. I've gone through fixation, I've gone through fixation, I've gone through reduction, and now I have 18 carbons. But if I'm going to finish out this cycle, I've got to get back down to 15 carbons. And so what will happen is that net gain of those 3 carbons, okay, so that gain of these 3 carbons, those 3 carbons are going to exit the cycle. Okay? They're going to exit the cycle, and they're going to be used to form half of a glucose molecule. You can see them here. Here's one of the G3P molecules. The Calvin cycle has been turning. It's made these G3P molecules. It's going to exit. Here's another one that exits from the Calvin cycle turning. Those G3P and pGal, those essentially quote-unquote extra G3P and pGal molecules that have exited, they're going to be combined to form my glucose. Okay? And so once they exit, right, when I got one of these guys that exit, now I get to go back down to 15 carbons. And so now that I'm back down at 15 carbons, those carbons will recombine, okay, and go back to forming the 5-carbon RUBP molecules. Okay, so let's look at the picture that kind of brings it all back together. This should be the same picture you guys have in your book. Okay, so you can see right here, this is the bottom of my chloroplast here. Okay, so just a reminder of where we're at. Okay, we're zoomed in the chloroplast. In particular, for all of this, we're in the stroma, right? We're not in the side, the thylakoid. We're not using the thylakoid membrane or anything. Okay, so I have these ribisco enzyme right here. Okay, so remember, here's my 5-carbon sugar right here, my ribulose bisphosphate, my RUBP. That's kind of the start of my cycle here. So I've got the RUBP, okay, and so that RUBP is going to combine with the CO2. So here's my CO2 coming into my cycle. Okay, so my RUBP and my CO2 combine. Remember, the ribisco is the enzyme that does that. Okay, so this is my enzyme that combines these two things together. Okay, and when those two combine, remember, I get my 6-carbon sugars, but those 6-carbon sugars are unstable, right? So those 6-carbon sugars pretty quickly during the reduction phase start breaking apart into 3-carbon, and you'll see here I've got 6 3-carbon molecules. You do not have to memorize all the names of these molecules, just so you know. I've got 6 3-carbon only the ones I've mentioned. Like, you don't have to know the name of that. You don't have to know the name of that. Okay, you just need to know the abbreviations. Okay, like I said, you don't have to memorize these names. So here I've got my 6 3-carbon sugars. So now I'm at 18 carbons. Here I had 3 5-carbon molecules, so I was at 15 carbons. Now I've got my gain. I'm up to 18 carbons. Okay, so the ATP is going to be used here. Okay, so this to reduce these, to reduce and rearrange. Okay, so I'm going to phosphorylate these 3-carbon sugars. So I've used some of the ATP that came from the light reactions. Then I'm going to reduce them. Okay, so you can see in my reduction here, molecule looks a little bit different. So I reduce them. Now I'm using the NADPH that came from the light reactions. So the light reactions have to be occurring for the Calvin cycle to work. And a lot of people call the Calvin cycle the dark reactions. Well, if they were the dark reactions, they were occurring when it was dark and the light reaction wasn't happening, I'm not getting any ATP, I'm not getting any NADPH, so this Calvin cycle isn't happening. So the Calvin cycle is happening at the same time as the light reaction. It needs the light reaction to power it. So I've got my 18 carbon molecules, I've got 18 carbons, okay, and so remember I've got extra, so essentially I have extra carbons. I've got to knock it back down to 15. Okay, so I get one of these G3Ps or P-gals, they're able to exit. Okay, so now I'm back down to 5 3-carbon molecules, so I'm at 15 carbons again. Using more ATP, I can arrange these 5 3-carbon molecules. I've got 15 carbons. Instead of having 5 3-carbon molecules, I can rearrange them now into 3 5-carbon molecules. I can get my RUBP back. I have to regenerate it to complete the cycle. Okay, just kind of, file this away, write your notes down, write your questions down. We'll, process whatever you need to process next class. Okay, so you've got your light reactions. So you've made your ATP, you've made your NADPH, and you've used that then to power your Calvin cycle, to actually start making the organic compounds. So let's start thinking experiment-wise. What are some things that could affect the rate of photosynthesis, and how could you test that in the first place? Okay, so some of the big things that could affect the rate of photosynthesis would be light intensity, okay, the availability of light and the quality of light that's available, okay, temperature. You've got an enzyme in that Calvin cycle reaction. Remember, enzymes are very dependent upon temperature. Okay, oxygen concentration also has an impact on the rate of photosynthesis. Okay, and so we're probably not going to do a photosynthesis experiment. We're definitely going to do a respiration experiment. But I do want you guys to be thinking in your heads, okay, how could I test this? How could you measure this, the rate of photosynthesis? How could you measure it in a lab? What would you be looking to measure, to gather data about, to determine if photosynthesis is even happening, so that you can graph some data and then calculate a rate? Okay, so let's look at light intensity, okay, because that was one of the factors that we just said could affect photosynthesis. So the rate of photosynthesis. So as a general rule, okay, as the intensity of light increases, my rate of photosynthesis is going to increase. So light intensity increases, okay, photosynthesis is going to increase, okay, up to a point. Okay, you see it gets up to a point. Now it doesn't decline and it doesn't stop. It just doesn't happen any faster. Okay, so it's not declining, it's not stopping. Okay, I just can't make the rate of photosynthesis happen any faster. Okay, and so it's going to increase here. Okay, so the increase in the intensity in light increases photosynthesis because you're increasing the excitation of those electrons in the photosystem. But eventually they become saturated. Okay, you reach the point of saturation which is a limiting level. They just can't get any more excited. Okay, and so it's not that they decline or anything. Okay, photosynthesis is now just occurring at a constant rate. Okay, we also said that temperature could affect the rate of photosynthesis. Okay, and so temperature can, as a general rule, as temperature increases, okay, photosynthesis increases, again, as you see on the graph, to a point. But this time it increases to a point and then decreases. Okay, and so if you think about what this is showing, okay, this is showing an optimum temperature. This is an optimum range curve here. Okay, I've got an optimum temperature right in this area, okay, of when photosynthesis will be at its fastest. And that goes back to the fact, again, that you've got this enzyme here. Remember, as temperature increases, I'm increasing the speed of my molecules. So my molecules are moving faster and they're colliding more frequently, and so my reactions will speed up because they're running into each other more often. Okay? The issue we have with temperature here, though, is that because there's an enzyme involved in the Calvin cycle, that ribisco enzyme that pairs up the RUBP with the carbon dioxide, that enzyme will denature when it reaches a certain temperature. Remember, the enzyme, their function is based on their shape. They're folded into that three-dimensional shape, and when they lose that three-dimensional conformation, then they don't work anymore. And so when the temperature gets too hot, the ribisco will denature, or it will lose its conformation, and it will be unable to do its job. Okay, and the last one that was mentioned was the concentration of oxygen. Okay, so O2 concentration. And if you look at your graph here, you'll notice as O2 concentration increases, photosynthesis actually decreases. Okay, so an increase in the concentration of oxygen. So essentially a buildup of oxygen inside of the leaf for some reason is going to decrease photosynthesis because it's going to cause something called photorespiration to happen. Okay, and we're going to talk about what that means in just a second. Okay, but that's why our rate of photosynthesis is going to decrease, because photorespiration is happening, and photorespiration, okay, is bad for the plant. And like I said, it decreases the rate of photosynthesis. So photorespiration occurs, like I said, when there's a high concentration of oxygen. So you've got your enzyme rubisco here, okay, that can join the CO2 to the RUBP. But if you'll notice here, rubisco can also join oxygen to RUBP. So when there's a high concentration of oxygen, okay, it will join that to the RUBP. So if there is more oxygen than CO2, so if the oxygen concentration is higher than the CO2 concentration, photorespiration will happen. And if photorespiration is happening, glucose is not being made. You are not able to do photosynthesis. Okay? And it requires, okay, it also uses the plant's ATP. So if that oxygen concentration dropped back down and the rubisco was now joining the CO2 to the RUBP, you still may not be able to do photosynthesis because the plant may have used up all of its ATP. So photorespiration, like I said, is bad for the plant. Prevents the synthesis of glucose and uses up the plant's ATP. This is mainly an issue for plants that are under what's called water stress. Okay, and so if you, we're going to jump into plants in the next couple lessons. If you just think about what you know about plants, okay, plants lose their, plants get water from their roots, and it travels up due to, adhesion and cohesion and capillary action and transpiration. Okay, you remember transpiration, where the plant is essentially the evaporation of the water from the leaves. And so if you think back to your leaf anatomy, okay, remember I've got my stoma or stomata. Okay, so remember your stoma were the stomata are those little holes on the underside of the leaf. Remember, the stomata allows for gas exchange. So it allows that CO2 to come in and the oxygen to leave, and it allows for water loss. That's how the water gets out during transpiration. So if a plant's under water stress, so it doesn't have access to a lot of water, it's in a dry, arid climate, it's going to close up those stomata because it's trying to keep water inside. So it's trying to prevent water loss. The problem is, though, when it closes up the stomata, now I'm affecting gas exchange. I'm limiting the amount of oxygen that's allowed out of the plant, and CO2 is not coming into the plant. So it's a trade-off. It's a compromise that the plant's making. To retain the water, it's hampering the gas exchange. And so the concentration of oxygen is going to go up, while the CO2 concentration is going to decrease. It's going to get used up, and it's not getting replaced. So those kinds of conditions, hot, arid, dry climates, those conditions favor this photorespiration process, okay, which is bad for the plant. So you've got certain kinds of plants that live in these areas through evolution have developed mechanisms to try to overcome this. Okay, so we're going to look at those now. Okay, so the first kind of plant we're going to talk about here is what's called a C3 plant. Okay, so a C3 plant, these are my normal plants. We're just going to review these for a second. Okay, so this is a normal plant. This is a plant that does the light reactions and the Calvin cycles here in the mesophyll of the leaf, like we talked about at the very beginning of the video. Okay, so light reactions and Calvin cycle happen in the same place at the same time. Okay, so they happen in the same place, okay, same time. Though both of those become an issue because both of those are things that other plants have developed ways around, where they'll separate the light reaction and the Calvin cycle, either physically separate them or temporally separate them by time. So C3 plants are our normal plants. Okay, they do light reaction in the Calvin cycle in the mesophyll of the leaves, same time, same place. So we've got what are called C4 plants that physically separate the two. Okay, so C4 plants, okay, examples of these would be things like sorghum, which if you've ever eaten that, to me is disgusting. Okay, sugarcane. Okay, so sugarcane, corn. Okay, all of these are what are called C4 plants. They thrive, they do well as a general rule in very hot environments. Okay, so a C4 plant does the Calvin cycle in a different place than the light reactions. Sorry for that complete blanking there. Okay, so the Calvin cycle and the light reactions are physically separated. They're taking place in two separate places. So the light reaction is still happening here in the mesophyll cells. Okay, so light reaction is happening there. The Calvin cycle, though, is happening here in what are called these bundle sheath cells. Okay, so there's my Calvin cycle. Okay, here's my light reactions. So these two are they're happening in two separate places. So the carbon dioxide that enters the plant, okay, so the carbon dioxide that's going in through the stoma, when the carbon dioxide is able to enter the plant, that carbon dioxide is shuttled to what's called to these bundle sheath cells. So the carbon dioxide, when it enters the plant, it's going to get transported to the bundle sheath. And the bundle sheath cells are impermeable to the carbon dioxide. So once it gets transferred in there, and we'll look at the pathway of how it gets in there, once it gets transferred in there, it can't escape and get back out. So it keeps the carbon dioxide levels high, because essentially the plant can, when it's cooler, like for example at night when the stoma are open, the plant can stockpile a bunch of carbon. Okay, it can pull the carbon in, and it can stockpile it into that those bundle sheath cells. So in the daytime, when the light reactions are happening, the stoma can be closed some to conserve water, and the light reactions, though, are still happening and can provide power to the Calvin cycle. So this is going to help favor the Calvin cycle over photorespiration. Okay, so the pathway the plant will use to get the carbon into those bundle sheath cells, okay, it's called the Hatch-Slack pathway. Okay, the Hatch-Slack pathway, it's going to happen before the Calvin cycle ever happens. Okay, you can see here's my CO2 entering the plant here. So the CO2 enters the plant, and that CO2 gets incorporated here. You can see this PEP carboxylase, so I've got an enzyme there, that PEP carboxylase. That CO2 is combined, okay, with my PEP molecule here, and the PEP carboxylase combines the CO2 with the PEP molecule. And now I have a 4-carbon molecule. This is where your C4 plant name comes from. Okay, so I've got this 3-carbon molecule of PEP here, combined with the 1 carbon from the CO2 using the PEP carboxylase. Now I have a 4-carbon molecule. Remember, I don't have to memorize all these molecule names here. But hopefully the names, what you will realize is that it did not use that Rubisco enzyme. Okay, remember that enzyme that didn't care whether it was carbon dioxide or oxygen, it would fix either one of them to the 3-carbon molecules. It uses this PEP carboxylase, okay, that will still fix the carbon even if the oxygen concentrations are relatively high. It will still fix the carbon. So I made this 4-carbon molecules, okay, and so this 4-carbon molecule will move through those plasmodesmata. Remember those holes, those gap junctions between the two plant cells? So it will move it through the plasmodesmata into the bundle sheath cells. So here I'm in the bundle sheath cell. Out here I was in a mesophyll, now I'm in the bundle sheath cell. Once it's into the bundle sheath cell, you'll see it releases that CO2 again. Okay? And when it releases that CO2, remember the bundle sheath cell is impermeable to CO2. So now that the CO2 is in it, the CO2 cannot get back out of that bundle sheath cell. And so the Calvin cycle, there's chloroplast inside the bundle sheath cell. The Calvin cycle is going to actually occur in the bundle sheath cell. This is ideal, like I said, for plants that require this, that may be under some kind of water stress. But it's not ideal for all plants because you'll notice it uses more ATP. I've got to convert this pyruvate molecule here back into the PEP, and that requires more ATP. So it's not favorable in that sense, as well as, remember when we're talking about evolutionary history, okay, the current plant is tied down by its evolutionary history. So if there was no mutation that made this beneficial, then it's just not going to happen for it. So that's your C4 plants. Okay, they physically separate the two. Right, so your CAM plants, okay, so what I have over my CAM plants, okay, they're not going to physically separate the two. They're going to it's going to be a temporal separation, which, if you remember, that's a time separation. Okay, so these are going to be a temporal separation here. Okay, and so they're open this stomata at night and obtain the CO2 and release the O2. And so it keeps them from drying out because they can keep their stomata closed during the daytime. Okay, back with your C4 plants, remember you used a different enzyme. You didn't use that Rubisco enzyme, so the stomata could be open a little bit and CO2 could be entering. And it didn't matter that there was a high concentration of oxygen because you're using a different enzyme. Okay, and then you transport that CO2 into the bundle sheath cell. In this case, the plant is opening its stomata at nighttime and collecting the CO2 at night. Okay, so you can see here, the CO2 gets stored at night, but it has to be used during the day. The Calvin cycle is not happening during the nighttime. You notice there's no Calvin cycle happening over here in the nighttime because there's no light reactions to power it. Remember, Calvin cycle's got to have the light reactions to power it. But so when the CO2 enters at night, okay, it's again converted into a 4-carbon molecule so that it can be released when the light reactions are occurring. So what I've got here is an overview of a plant that would do this. You can see this cactus here. Those would be some examples. Succulents, things, with a big, heavy, thick, waxy cuticle, aloe, cacti. They would do their CAM plants. Pineapples are CAM plants. Okay, so just some examples of plants that can do this. Okay, so I've got my CAM plants here at nighttime opening up the stomata, allowing CO2 to come in. Humidity's a little higher, temperature's a little lower, they're not as likely to dry out. Okay, so the CAM plant then closes that stomata in the daytime, okay, but the CO2 was stored. So at night, the CAM plants have combined the PEP, okay, combined with that PEP molecule to form the C4 acid. So I've got a 4-carbon molecule here. And in the daytime, that 4-carbon molecule gets broken down to release the CO2. And so now the Calvin cycle can occur right here, being powered by the light reactions that are occurring during the daytime. Okay, so just kind of a little review of the two. Okay, over here on the left, I've got my C4 plants. On the right, I've got my CAM plants. Remember my C4 plants? Okay, I've got a spatial or physical separation here. They're happening in 2 different places. Okay, both of them, okay, so they both are incorporating it into a 4-carbon. They're incorporating the CO2 into a 4-carbon organic acid. They're doing that carbon fixation. Just the CAM plant does it at nighttime. Okay, the C4 plant does it in a different space. Okay, it's doing it in the mesophyll cell. Okay, and then both of them, they'll use that organic acid to release the CO2 when the Calvin cycle can happen, which temporally, remember, is going to happen in the daytime for the CAM plant, so I have my temporal separation here of night and day. Okay, and then it's going to happen in the bundle sheath cell of the C4 plant, so I have my physical space, my spatial separation of the two. Okay, so that's kind of the end of our photosynthesis. We've got Calvin cycles, so we're forming the actual organic compound, as well as some of these adaptations that plants have done to overcome the photorespiration so that they can encourage photosynthesis to happen. So remember, don't forget to be thinking about how we could measure in a lab whether photosynthesis is occurring.